I want you to try a little mental exercise with me, if you can, just for a second. Imagine your body not as, a biological thing, but as a city, a massive, sprawling metropolis.
And not a quiet one either. We're talking about a city with billions of citizens all interacting constantly.
Exactly.
Yeah.
It's just chaos. You have traffic lights changing every millisecond, road crews patching potholes in real time, delivery trucks flying down highways. It's kind of a miracle it all works. But here's the scary part. What happens when we try to map all that chaos? Or even harder, what if we try to predict when one single traffic light, one tiny genetic switch is about to go out?
Well, historically, we don't predict it. We just wait for the crash. We see the pile up on the highway, and only then do we go back and try to figure out, which light failed.
But that's the old way. Today, we're doing a deep dive into research that suggests we're moving out of that era of just observing and into an era of calculation.
It's a huge shift from biology as a descriptive science to biology as a predictive, almost an engineering discipline. And we're looking at it through two really different lenses today.
First up, we have what I'm calling the hardware side of things. It's a new database. It's called PEPBI. That's PEPBI from researchers at Auburn and Georgia Tech.
Right.
And it's basically a Rosetta Stone for how proteins and peptides physically shake hands.
Which, I know, it sounds super niche, but that handshake is the absolute foundation of modern drug design. If you don't get the handshake right, you can't make the deal.
And then second, we have the software side. A huge paper from Nature Genetics introducing something called POP-EVE. And this is an AI that combines millions of years of evolution with modern human population data.
And it uses that combo to spot dangerous genetic typos that other models they completely miss them.
And not just spot them, right? It scores them.
It scores them. It tells you exactly how bad a mutation is compared to every other possible mutation in your body. It's the first time we've ever had a proteome-wide scoreboard.
So our mission today is to figure out how we get from wet labs and petri dishes to these like AI detectives and molecular matchmaking. How are scientists using these tools to build new drugs and diagnose rare diseases faster than we ever thought possible?
Because these really aren't just cool new toys. These tools are finally solving these fundamental data gaps that have held back precision medicine for decades.
Okay, let's get into it. We'll start with the hardware, the physical stuff. We're talking PEPBI and the world of peptides. Now, before we get into the database itself, can you give us the explain like I'm 5 on what a peptide actually is?
Simplest way to think about it. Imagine a protein is a whole novel. It's a long, complex chain of amino acids, all folded up into this massive functional shape.
Okay.
A peptide, that's just a sentence from the book. It's a short chain, maybe 5 to 20 amino acids long in this context.
So they're small, but important.
Incredibly important. Because they're small, they can be messengers, they act as hormones, structural bits. They're basically the keys that float around the body and unlock specific functions in those big proteins.
Right, so if they're the keys, that makes sense why we use them for drugs. If I want to shut down a cancer signal, I just need to design a peptide key that fits that lock and jams it.
Precisely. We use them for therapeutics, for diagnostics, all the time. But here's the problem that has just plagued biochemists for, 50 years. We know what the key should look like, but designing one that actually works, it's just incredibly hard.
But why? If I have the blueprint for the lock, why can't I just cut a key to fit?
Because biology isn't a static thing. It's dynamic. It's always moving. The real bottleneck has been a lack of what we call paired data.
Paired data.
Yeah, so usually in our databases, we'll have the structure like a 3D snapshot of what the molecule looks like. Or in a completely separate database, we might have the thermodynamics numbers on how strong the bond is.
But you rarely get both for the same handshake.
Almost never. And without both, it's like trying to design a key when you know the shape of the lock, but you have no idea if the metal you're using is too soft to actually turn the bolt.
That's a great analogy. The key looks perfect, but then it just snaps off in the lock.
Exactly. So this is where PEPBI comes in, this new database.
From the Auburn and Georgia Tech team, what makes it so different?
It's the curation. It's incredibly rigorous. PEPBI has 329 peptide protein complexes. And look, I know 329 sounds like a tiny number in the age of big data.
Yeah, I was going to say, that sounds like a slow Tuesday afternoon for Google.
Right. But in structural biology, this is an absolute goldmine because of how strict their criteria were. They didn't just scrape data. They selected these complexes based on 8 different criteria.
Like what?
Well, for one, every single structure had to have a resolution of 2.0 angstroms or better.
And for those of us who don't have a microscope handy, What does that mean?
It means you can see individual atoms. It is crystal clear. But the real game changer, and this is what makes PEPBI so special, is that for every single one of these, they also found a known unbound structure.
Unbound. You mean what the protein looks like before the key goes in?
Exactly. And that's crucial. You have to see how the pocket, the lock, changes its shape when the key enters. Most databases just give you the after photo. PEPBI gives you the before and after.
So it actually captures the movement.
It captures the induced fit, but then they went even further. They included the thermodynamics, Gibbs free energy, enthalpy, and the big one, entropy.
Okay, entropy. I have this vague memory from high school physics. That's chaos, disorder.
In a sense, yes. And this is the silent killer in drug design. You see peptides floating around in your blood, they're naturally disordered, they're floppy, they just wiggle around constantly.
Like a wet noodle.
A perfect analogy. But when that peptide binds to a protein, it often has to freeze into one specific rigid shape. And that transition from floppy to rigid, it costs energy.
Wait, so the peptide has to pay an energy tax just to stop wiggling?
It's called the entropic penalty. It's giving up its freedom. And if the energy it gets from binding to the protein isn't stronger than the energy it costs to stop that wiggle, the drug won't stick. It'll just fall right off.
And our current computer models, they don't account for the wiggle.
They're terrible at it. Because we've never had the right training data. We've just been guessing at the cost of the wiggle. But PEPBI provides the hard numbers for that entropic cost for the first time.
I see. So you feed this super curated database into an AI, and you can train it to understand not just if the key fits, but if the key is willing to pay the price to stay inside the lock.
That is the perfect way to say it. They even use this tool, the Rosetta Interface Analyzer, to calculate 40 different binding properties for each one. So now an AI can learn the physics of what makes a good, tight bond.
That is just huge for designing drugs. You stop hoping it works, you start calculating that it will work.
Exactly. And that really covers the molecular level. That's the handshake. But now I think we need to zoom out, because the second paper deals with the blueprint itself.
Right. So we know how things bind. But what happens when the genetic code has a typo? We're shifting over to that Nature Genetics paper and this new AI, PEPBI.
And this moves us from designing molecules to diagnosing people. It tackles one of the most frustrating problems in modern genetics, the variant of uncertain significance, a VUS.
I have heard that term. It basically sounds like a doctor shrugging their shoulders.
That's pretty much what it is. A doctor sequences your DNA, finds a mutation, and has to say, look, we found something, but we have absolutely no idea if it's dangerous or not.
So you don't know if it's a harmless quirk, like having attached earlobes, or if it's a deadly tycho.
Precisely. And until now, we've had some AI models to help. There was a model called EVE, just EVE, that was pretty good at ranking mutations, but only within a single gene.
Okay, so it could look at gene A and tell you this specific mutation is the worst possible thing that can happen to gene A.
Right. But it couldn't tell you if the worst thing for gene A was more or less dangerous than a medium bad thing for gene B. It lacked a universal scale.
Back to our city analogy. It's like EVE could tell you about the worst traffic jam in a tiny village and the worst traffic jam in New York City.
Yes, but it had no way of knowing which one was actually more disruptive to the whole country. The village jam is severe for the village, but the NYC jam could crash the entire system. Previous models couldn't tell the difference.
So enter PEPBI. And the POP is for population, right?
Yes. This model is much smarter because it combines two gigantic data sets. First, it uses deep evolution. It looks at millions of years of data across all of life to see what nature allows and what it forbids.
So if a monkey, a mouse, and a fish all have the exact same protein sequence at one spot, and you have a mutation there...
Then your mutation is probably very bad. Evolution has protected that spot for a reason, so that's ingredient 1. But PEPBI adds ingredient 2, human population data.
And this is where things like the UK Biobank come in. Real genetic data from hundreds of thousands of living people.
Correct. And it uses a really clever statistical method called a Gaussian process to combine the two.
Okay, Gaussian process sounds complicated. What is it actually doing with the data?
Think of it like a calibration filter. It uses all that modern human data to fine-tune the ancient evolutionary data. Essentially, it looks for the empty spaces.
The empty spaces.
Yeah, imagine a mutation that evolution says might be okay. Maybe some fish have it. But then you look at the data from 500,000 healthy, living humans, and not a single one of them has it.
The dog that didn't bark.
Exactly. The model learns that for humans, that mutation is forbidden. If 500,000 people are walking around today and none of them have that typo, it strongly implies that the typo prevents you from walking around.
That's a bit chilling, but I see how powerful that is. So what does this let POP-EE do that other models couldn't?
It creates a severity spectrum. Most AIs just give you a binary answer, pathogenic or benign, bad or good. POP-EV can actually distinguish between mutations that might cause an adult onset disease versus those that are so severe they cause death in childhood.
That is a huge, huge distinction for a doctor or for a family.
It places every single protein on the same scale of importance, and they've already proved it works. They ran POP-EV on a group of patients with severe developmental disorders.
And these were kids who were already undiagnosed, right?
Yes, the standard genetic tests had come up empty. But POP-EV went through their genomes and flagged 123 novel candidate genes for causing their disease.
123. That's not a small number in genetics.
It's enormous. These were genes that doctors had maybe suspected but could never prove were the cause. POP-EV flagged them with incredibly high confidence.
Can you give us a concrete example?
Sure, let's look at a gene called ETF1. This gene is part of the ribosome, the actual machine in your cells that builds all the other proteins.
So pretty important machinery.
The most important. POP-EV flagged 2 specific variants in ETF1 as being extremely damaging. But here's where the 2 parts of our deep dive today, they collide. Does POP-EV know why they're bad?
Well, it shouldn't, right? It's just a statistical model. It's looking at population counts, not physics.
That's what you'd think. But when the researchers zoomed in on the 3D structures of these new disease candidates, they found something incredible. 91% of the damaging variants that POP-EVE found were within 8 angstroms of a critical interaction site.
8 angstroms. In molecular terms, that's basically touching it.
It's a direct hit. They saw this over and over. In another gene, HDAC2, which helps package your DNA, POP-EVE found a variant sitting right in the active site, right in the engine of the protein.
So the AI found the most important functional parts of these proteins just by looking at population data.
It deduced the physics without being taught the physics. It proved that the statistical signal, the missing people in the database, was pointing directly to a real physical breakage in the molecular machine.
That's incredible. It's like finding the smoking gun without ever having to visit the crime scene.
And it does it for the whole genome at once.
There's one more feature here that I think is the real breakthrough for families. The paper calls it the no parents needed feature.
Yes, this is the part that could be a clinical revolution.
Okay, so normally if a child has a rare disease, the gold standard is trio sequencing. You sequence the child, the mom, and the dad.
Right. You do that to find what are called de novo mutations, brand new typos that happened in the child and weren't inherited. It's basically a subtraction game. You take the child's DNA, subtract out the parents, and see what's left.
But that's expensive.
Yeah.
And sometimes parents just aren't available.
Exactly. What if you're an adult patient? Or you were adopted? Or one parent is unavailable? You can't do trio sequencing. And without it, finding the causal gene goes from finding a needle in a haystack to finding a needle in a stack of needles.
So how does POP-EV get around this?
Because POP-EV is calibrated so precisely against the general human population, it's accurate enough to find the causal mutation in the child without parental data.
It doesn't need to do the subtraction game.
No. They tested this. In cases where they knew a de novo mutation was the cause, POP-EV ranked that exact mutation as the #1 most damaging variant in the entire genome in 98% of cases.
98%.
98%. It pointed to the single typo out of millions and said, that one. It's that one. All without needing the parents for comparison.
That could open the door for so many singleton patients who just hit a dead end. That's a lifeline.
It really is. It could democratize genetic diagnosis. You don't need a perfect family unit or 3 times the budget to finally get an answer.
Okay, so let's tie this all together. We started with PEPBI, giving us the raw physics, the thermodynamics of how molecules actually stick to each other.
The hardware specs, you could say. Understanding the entropy, the wiggle, the binding energy, all of it.
And we end with POP-EE, which uses evolution and population data to give us a fitness score for the entire human proteome.
The operating system integrity check.
It really feels like these two worlds are converging. We're moving away from a world where we just guess at biology. We're starting to compute it.
From observation to calculation, with PEPBI, we can start designing drugs that we know will bind tightly. With PEPBI, we can pinpoint the cause of a disease with a level of certainty we've never had.
It's high-fidelity biology.
And it's only going to get more powerful as we sequence more people and solve more 3D structures.
Before we wrap up, there was one final thought in that POP-B paper that really stuck with me.
Yeah.
It was, well, it was a little haunting.
I think I know what you're talking about, the invisible landscape.
Yes. The model predicts that there are many, many genes out there capable of causing incredibly severe diseases that we've never actually seen in a patient.
Right. The model looks at a gene and says, if this one breaks, it would be catastrophic. But then we look through all of our patient databases, even the biggest ones, and there's no one.
So the question is, are there thousands of these invisible genetic diseases out there just waiting to be discovered?
Are those mutations so severe that a pregnancy never even comes to term, that life never begins?
It makes you wonder what we're not seeing. Are these the silent tragedies behind miscarriages or infertility? Or are they just rare conditions so unique we haven't connected the dots?
It suggests there's this entire invisible landscape of human biology that the AI has started to map, and we're just now beginning to explore it.
It's a humbling thought. The map is getting sharper, the resolution is getting better, but the territory is still full of mysteries.
And that is exactly why we do this.
Well, thank you for walking us through the molecular city today.
It's always a pleasure to navigate the traffic with you.
And to you listening, thanks for diving deep with us. Whether you're a peptide looking for a thermodynamic match or just trying to avoid a genetic traffic jam, stay curious. We'll see you next time.